As you're all aware, stem cell therapies are a very active clinical trial space in regenerative medicine. And after now quite a number of years of multiple kinds of stem cell therapies going on in the eye, we have an idea that the regenerative effects of the stem cells are not always the full story. But nonetheless, the paracrine effects of stem cells are also thought about in a very limited way. They are mostly thought about in terms of just growth factors. So the talk today is about non-regenerative factors in regenerative medicine, or paracrine effect stem cells. So the idea that the eye was active in the early clinical trials for stem cells was not a surprise, because the eye is just relatively ideal compared to other sites for transplantation. The small graft size means that the manufacture of the graft is relatively inexpensive. So 100,000 cells for a macular degeneration therapy is not a crazy number of cells to think about, but the billions of cells that you would need to build a liver, for example, just dwarf that. There's very easy visualization of the disease process with all of the wonderful imaging tools that have come through ophthalmology, and of the graft itself, so that if a teratoma should result from a pluripotent stem cell therapy, its removal, even with enucleation, should not kill the patient. From my perspective, the eye is really an important place for understanding how stem cell therapies are working in the CNS and in the spinal cord. Once you put cells into the spinal cord, they go into a black box. And since developmentally the eye is very similar to the brain, these therapies really should inform the CNS therapies. So here are some broad stroke generalizations that come from thinking about stem cell therapies in multiple organ systems. The first is, why did we get stuck with paracrine therapies being equal to trophic factors? I think the answer is because early on it was developmental biologists who entered this field, and the paradigm for regeneration became to recapitulate normal developmental processes, and people talked about trophic factors, growth factors. But what we've found out both in preclinical and in clinical models is that if you take one of those apparently switch-like trophic factors from development and try to use it as a therapy in an adult human, in general, these are not very effective when given alone. So you are more aware than I am of the GDNF story. It's not probably going to be the whole answer to treating macular degeneration. And interestingly, the Neurotech CNTF trial, which has been listed as complete in phase 2 on clinicaltrials.gov, is being delivered by a cell. So the cells have been manipulated to overexpress the growth factor, but perhaps they are secreting other factors, so you're not really given a single trophic factor here. So likely more than one trophic factor is going to be much better than a single trophic factor, and that means that cells will likely be the source of trophic factors since they secrete many of them, and it just would be too hard to package independent trophic factors otherwise. For the eye, we also have some very interesting clinical studies going on in parallel to cell therapies that we should try to compare to the cell therapies for potency and perhaps for mechanism. So it's going to be very interesting to see whether transgenic cells that overexpress a growth factor are more effective than the single trophic factors, but whether they're more effective than unmanipulated cells is also an important question, and whether potential gene therapy approaches to these trophic pathways will be a better approach than any cellular therapy. So I'm not saying that the trophic factors are unimportant. They are, and they're complicated. So in the case of mesenchymal stem cells, which are being used in clinical trials for AMD now, there's a whole host of factors that these cells can secrete. And fortunately, most of the growth factors are neuroprotective, or a lot of them are neuroprotective, including VEGF and PDGF, CNTF, GDNF, and BNDF. So these are complicated. And you should also know that one company's or one lab's mesenchymal cell is not necessarily the same as the others. We are recognizing that these cells are not well characterized and that they're quite heterogeneous in terms of their expression profiles. And this is something that will sort itself out along the way. So in looking at the neurotrophic factor profile for cells that have been used for AMD or are likely to be used in the eye, there are some common growth factors that you see secreted. NGF, BDNF, and NT3 seem to be commonly secreted by these cells. But interestingly, VEGF and FGF2, both angiogenic, are secreted by some of these populations as well. And it's important to note that NGF across different systems and disease models is also angiogenic. Do we really want angiogenic factors secreted in the context of AMD, even though they are neuroprotective? Retinal stem cells and the pluripotent cell therapies for generating RPE, the profiles for these cells are largely unpublished because they're proprietary and the companies have held this pretty close to the chest. But nonetheless, I was able to find evidence that the human-induced pluripotent stem cells being generated to differentiate RPE do secrete VEGF. And then just for the sake of completeness, it's important that trophic factors that are not traditionally associated with the eye be considered in developing therapies down the road, including erythropoietin, which is globally neuroprotective and has been used in clinical trials for ischemic optic neuropathy. More recently, progranulin really seems to have some protective effects. This is a growth factor that was derived in animal models from adipose-derived stem cells. So stepping away from the growth factors, another way that a paracrine effect could be mediated is through the control of vascular tone. And this slide summarizes some work from my lab at Emory outside of the eye. We were using a pain model that involved chronic constriction injury of the sciatic nerve of the rat. And then we're giving bone marrow mononuclear cells, a mixed bag of stem cells, to see what happened with the pain behaviors. And these cells turned out to be very potent in terms of reversing the pain behavior, more potent than drugs that had been tried in the same model and then went on to be used clinically for neuropathic pain. So thinking back on developmental biology, we decided that the heat shock proteins, which are important in development of the peripheral nerve, might be mediating some of the effect. In particular, heat shock protein 27, very important for peripheral nerve development, was what we expected to go up as a consequence of the cell therapy. However, it didn't. But heat shock protein 32 did. And heat shock protein 32 is the source of carbon monoxide, a potent vasodilator. That made us think about this vascular tone issue, and we started to look as well at the endothelin receptor axis and found that there was relative downregulation of endothelin A, signaling through endothelin A mediating vasoconstriction, versus relative upregulation of endothelin B receptor mediating vasodilatation. So there were two pretty potent cues that probably allow some control over local vascular tone, and you can imagine in models like MI and stroke, which are largely ischemic, or in this kind of constriction model, which certainly has an ischemic component, that the cells could be mediating a therapeutic effect through this particular paracrine mechanism. Another way that the cells can affect their environment is by manipulating the immune cells around them. We know in the eye that limbal mesenchymal stem cells suppress T cell proliferation. Suppression of T cell proliferation is the kind of common mechanism that mesenchymal type stem cells or multipotent adult progenitor stem cells use to manipulate the immune environment. And again, if you look in clinicaltrials.gov, MSCs are being used for hundreds of different diseases because of the common inflammatory drivers of a lot of disease. And they're also being used for lots of autoimmune disease, in part because of this observation that they suppress T cell proliferation. The mesenchymal stem cells are certainly reaching clinical trials for AMD as well. And I think the anti-inflammatory factors are likely to be very important in their therapeutic effect. But the way the trials are designed, there are no immunologic endpoints. And so we really won't get insight into the anti-inflammatory mechanisms in these early clinical trials. The other way mesenchymal stem cells can change the immune environment, and this is very important, is that they can, in multiple organ systems, including the spleen, if the cells are given IV, promote a phenotype switch of macrophages from a pro-inflammatory phenotype to an anti-inflammatory phenotype. Now, with Jerry Luddy's work characterizing the local inflammatory cells, there seems an opportunity in the animal models of disease to address some of these immunosuppressive properties in treating macular degeneration. What's clear when you look at the paracrine effects, the non-regenerative effects of cell therapies, is that temporally these effects outlast the residence time of the cells. So that really makes us think about invoking other mechanisms besides growth factors, which could only be secreted locally if the cells are still there. So particularly MSCs are cleared very rapidly, whether they're given IV and home to the site of injury or whether they're given intraparenchymally. They're certainly gone within weeks, often within days. An interesting exception might be the vitreous. I found a paper in which intravitreal injection of MSCs resulted in them just kind of hanging out in the vitreous. They don't proliferate there, but they survive for a long period of time. They apparently don't have an escape route if you put them in the vitreous. Nonetheless, this temporal discrepancy between the long-lasting therapeutic effects and the residence time of the cells really suggests to me that there's a hit-and-run mechanism that we should be thinking about, or mechanisms. Sally Temple's paper, which is not yet available in its full form, in which she describes a new progenitor for retinal pigment epithelial cells, apparently suggests that they work after transplantation by promoting what's left of the endogenous regeneration mechanisms in the eye. You can also promote endogenous regeneration by calling progenitor cells to the area, and this has been a mechanism that's shown by Stephanie Dimler in the heart, where activation of resident progenitor cells in a distant site from the actual graft call in the endogenous repair mechanisms. To say that the microRNAs have surprising function. So we don't want to be fooled into thinking about paracrine effects as just growth factors, but we also don't want to be fooled into thinking about microRNAs as mediating the same function as the cells themselves. So, for example, in one company's data that I've seen in some detail, and in other literature emerging, a cell therapy that has a protective effect in a chronic stroke model, for example, if you take the exosomes and the microRNAs and do the same experiments, there's not a protective effect in terms of the stroke, but the microRNAs have a surprising anti-cancer signaling signature. And this anti-cancer signaling signature, I think, is emerging over and over again and may well be an important part of immune surveillance of exosomes for cancers that is, again, something at the very beginning of our understanding. In terms of the eye specifically, and microRNA profiles, I was able to see that people are thinking about these profiles as markers of the stage of disease for AMD, but again, very beginning kind of trials, understanding. So exosomes are taken up actively by the cells, and no one has really looked at how long they are active inside a cell that has taken them up versus, for example, the residence time of MSCs. But it makes sense that if you're initiating transcriptional changes in the cells with microRNAs, plus all the other potential signaling pathways that happen inside the host cell, that you could have a prolonged therapeutic effect mediated by exosome internalization that outlasts the residence time of the cells. So exosomes and endosomes are well known in some systems to be highly regulated, and this is just an example of the transferrin life cycle in which exosomes and endosomes are very important. But as I said, the CSF is loaded with these things. The vitreous certainly has them as well. Vitreous is spelled wrong. And we also know that other components and other cell types secrete specific populations of exosomes. So preclinically, people are starting to think about this. There's nothing in clinical trials, as far as I know, with exosomes, but I think it's coming, and it's coming soon. They are anticipated to be used alone or in combination with cells. The alone is particularly appealing if you could recapitulate what a pluripotent derived cell therapy does without the danger of the cell itself. They are given into parenchyma or into circulation, or linked to engineered gels and fibrin spun threads in order to be able to time their release. Looking specifically for how this relates to AMD, I stumbled across a paper by Jim Handa, and at the meeting you can ask him about this. But it seems that he's discovered an exosome release pathway that affects the complement regulatory factors. And so it raises the issue in the case of macular degeneration about whether exosomes can be used to modulate the complement component of the pathophysiology. And we're now, again, understanding that each cell type seems to regulate its own exosome secretion and quality, quantity, and heterogeneity. Specific exosomes have been found in aqueous humor. Are they the same as in vitreous? I don't know. Retinal astrocytes secrete their own exosomes, and retinal pigment epithelial cells secrete exosomes that contain alpha crystalline. For AMD, perhaps these can be thought of as useful biomarkers down the road for both the presence and stage of disease. The second hit-and-run mechanism that potentially long outlasts the residence of the grafted cells is mitochondrial transfer. And unfortunately, the seminal paper in this field does not have very good images, so I'll try to walk you through this a little bit. A lab at Columbia, Bhattacharya's lab at Columbia, induced an acute lung injury model in mice by injecting lipopolysaccharide, the bacterial endotoxin. And the mice were treated with human bone marrow-derived mesenchymal cells given down the mouse trachea. They noted that the graft cells formed bridges with the alveolar cells. And the bridges were formed by connexin 43. Connexin 43, of course, is the protein that mediates the cell-cell connections in the cardiomyocytes. And you can see a little bit of how this was demonstrated with the alveolar type 2 cells labeled in green and the donor human mesenchymal cells labeled in red, and the yellow, of course, is the overlap. Through these connexin 43 bridges, they were able to image mitochondria traversing, but they don't have a very good picture of that. Nonetheless, the mitochondrial traversing meant that the cells that were in the equivalent of the penumbra, the cells in danger of dying because of this LPS toxicity, increased their mitochondrial function, increased the production of ATP, and they were more likely to survive than untreated cells. So mitochondrial transfer may well be a very potent trophic effect. Certainly this was anti-apoptotic, and the energetics of the host cells that received these mitochondria suggest that this may be a very important mechanism for us to think about in diseases, especially where there's hypotension or ischemia. So I'm hoping that this will spur a little conversation at the meeting about what exactly is going on when we put cells into the eye. Fortunately, we don't have to have any detailed understanding of the mechanism of action of cell therapies before we enter clinical trials. Otherwise, none of these would be going on right now. But we have to think about these complex paracrine effects. Just like small RNAs were missed when the genome was sequenced, we've really missed the boat in terms of how important exosomes are in cell-cell communication and potentially in immune surveillance. Mitochondrial transfer, I think, now that we are getting tools to examine mitochondrial energetics in traditional labs, will also be important as a paracrine effect of cell therapies. And understanding these mechanisms will really help us dissect the therapeutic effects of the cells that we're giving out of this complex soup of potential therapeutic mechanisms. Once again, I think it's really critical that since so many different kinds of cell therapies are happening in the eye, that we think mechanistically as we compare the results that come out of clinical trials. Will the regenerative RPE given for AMD be as potent as these combined paracrine cargoes? It's hard to say at this point. Nonetheless, understanding the mechanism will be important for us in designing future therapies that have nothing to do with cells or in optimizing our cell therapies for AMD.